So let me just get to the point. The other talks have set this up really well. My disclosure is I only work with one big drug company, which is Eli Lilly, and I'm going to focus on the material I know the best, which is about Solanezumab in particular. But what I did want to do is emphasize a couple of points about, first of all, drug development. Now, there was a comment made earlier today about translational research not being very interesting or whatnot. My apologies, but that's actually been a focus for us. And the other point that really is in keeping with the theme of the meeting is to talk a little bit about where we are as a field in terms of earlier treatments, prevention of symptoms, or delaying or slowing progression. So it's really a question of maintaining health as much as it is treating disease symptoms. So let me just talk a little bit about here. You've already heard a little bit about amyloid and tau. I don't want to get into a big debate between the Baptists and the Taoists here. I think in the view of some of us is that many, in fact, the majority of the genetic mutations actually look at the amyloid and A-beta pathway. Tau mutations actually cause other diseases, but they're both good targets. Our thinking has just evolved in terms of amyloid and A-beta perhaps being upstream, tau being a downstream effect, and that tau effect is actually closer to clinical symptoms and neurodegeneration. So it's certainly not to say that they're not both good targets, and as we'll talk about a little bit, it may be just when you use particular treatments to go after those targets. So if you go to the next slide. So this is a slide that Risa Sperling was kind enough to lend to me. If you hit it again, this shows the prevalence, the age-dependent prevalence of Alzheimer's disease. The biggest risk for Alzheimer's disease is your age. If you hit it again, this shows pathological data that's really been available for a number of years that shows that if you look at asymptomatic people who die, that's the prevalence of having plaques in your brain that are asymptomatic. And there's always been this question of, is there this lag time between when you develop plaques and then when you develop symptoms? Now, if you hit it again, what PET scanning and these amyloid ligands have allowed us to realize is that this does, in fact, seem to be the case. If you look at asymptomatic people in the presence of amyloid plaques based now on PET scans rather than pathology, you see exactly what the pathology showed. And if you hit it one more time, if you could advance it one more time, so this creates a real opportunity. So now we have this 15-year window between the time when you first develop the amyloid pathology and when you start to develop symptoms. And that's something that I think we can really use to our advantage. So if you advance again. So this is a slide. It's a little unusual to be the last Alzheimer's speaker and be the first person to show this slide, but it's a slide that was developed by Cliff Jack and a number of others. And this is a hypothetical slide that looks at what a lot of the current thinking is in terms of the sequence of what happens with disease pathology. So I wouldn't get too caught up in exactly when the lines start to move specifically in relation to each other, but the current thinking then is that the first step is the deposition of amyloid plaques. That's followed then by neuronal dysfunction, changes in FDG, changes in fMRI. Tau probably starts to accumulate at about the same time. It's not till a little bit after that you develop enough neurodegeneration that you can pick up atrophy on an MRI. And then finally, after all of that, is when you first start to develop changes in cognition. After changes in cognition in a stage we sometimes call mild cognitive impairment, it's only following that that you eventually develop changes in your function, in your activities of daily living. So there's a sequence of events here that seems to play out. The ADNI data, this is a large observational study, seems to show very similar sorts of results. But again, this also creates some opportunities. So one of the things that's been mentioned before is primary prevention. Now, primary prevention is great when you're talking about infectious diseases. So if you want to vaccinate against a virus, that's great. But if you want to vaccinate against an endogenous protein or peptide, whether it's A-beta or tau, then the risk-benefit ratio is a little less clear. So I think it's certainly a noble cause, and from a public health standpoint, it would be terrific. But I think right now, If you want to do primary prevention, what you need is a good Mediterranean diet, some red wine at night, go out and get some exercise. All the things that help your heart will probably help your brain, and there's actually some pretty good evidence for that. But if you hit it again, but where a lot of work is starting to go on now is secondary prevention. So again, this window of opportunity between the time when you start to get plaques but before you've developed any symptoms. And I'll come back and talk about that just a bit more, but that creates a real opportunity. And then finally, if you hit it one more time, we get into our tertiary prevention, if you want to think of it that way. But these are people who have started to develop symptoms, at least mild cognitive impairment, and now we're trying to slow down the rate of clinical decline in those measures. So if you go to the next slide. So what we've really come to here is a continuum of the disease. And so you start off with plaques and no symptoms. You end up with people with dementia, which is at first mild, then moderate and severe. And as I'll kind of walk through a little bit here, at least some of the current thinking is that these amyloid-related therapies may be best for people with at least mild AD dementia. That may not be too late, and before that, maybe even into moderate a little bit. But maybe the more moderate and severe and maybe even the mild dementia patients are really more appropriate for tau therapies. I mean, that's part of the current thinking right now. Go to the next slide. So a lot's been said about these various failures of Alzheimer's disease treatments. And the top half of that slide shows you the symptomatic treatments that have been successful. As Rochelle mentioned, these things do have modest effects. We wish they were better, but there are well-characterized effects of these drugs. When you try to actually slow down underlying disease pathology through completely different mechanisms, these are not neurotransmitter-based, it's been a little tougher road to go. Now, as I'll come to at the end, I think we actually are making some progress. We have learned some things, but there's been a fair amount said about the number of shots on goal that people have taken, and so far haven't had any clear successes. But let me dig into that just a little bit more. So if you go to the next slide. So this is a slide that was from July 2011. So it's out of date now, but it's to make a point. So at that time, these are the drugs that had been looked at for possible disease modification in Alzheimer's disease. Now, if you hit it again, now some of those were amyloid-based, and for some of those, the mechanism wasn't quite as clear, and won't go into the details. For the ones that didn't have a clear mechanism, there was just no effect one way or another on cognition. If you hit it again, the drugs that were amyloid-based, now some of those drugs didn't really have really clear evidence of target engagement in the brain at the dose that was used in the phase 3 trials. So this is the part of drug development that tends to get skipped over a little bit. There are some good reasons for it, and tramaprosate in particular, there was a little bit of data there, but generally not a lot of data that it got into the brain at the dose in phase 3 had no effect. There was one drug that was one that I was involved with, semagacistat, this gamma secretase inhibitor that you heard about. We did have good evidence of target engagement, both peripherally and more importantly in the brain. We really thought at the dose we took into phase 3, we inhibited the gamma secretase enzyme. And in fact, it did have an effect on cognition. Unfortunately, it wasn't the effect that we wanted. It actually made cognition a little bit worse. We stopped the trials prematurely because of that. What we have found out before or since getting into these phase 3 trials is gamma secretase, that enzyme, does a lot of things, and apparently it does a lot of things in the brain more than just making A beta. So we inhibited the enzyme, it made less A beta, but apparently that enzyme also does some things that your brain needs. And so when you inhibited it at the doses that we used in phase 3, the bad things outweighed the good things. Now, it's still a little hard to tell this story, but at the time especially, I think the important point was you had to learn from this, that this was the first time that based on good evidence of target engagement in the brain, you actually had an effect on cognition. So it wasn't what we wanted to have happen, but there was something that you could learn from. So if you go to the next slide. So that takes us then to Solanezumab, and let me show you one cartoon just to talk a little bit about the mechanism of how it works. I'm not sure how this is going to go doing it from the back, but if you hit it again, Here you see beta secretase and gamma secretase cleaving APP. It forms the little yellow A betas. A betas are trafficked out of the brain very rapidly, cleared by the liver. In the case of Alzheimer's disease, in sporadic disease, we think the clearance isn't quite fast enough. It accumulates. It causes these plaques to form, and a lot of things happen then downstream of the plaques is our current thinking. So if you advance it again, so if we add our monoclonal antibody, in this case solanezumab, it binds to the A beta in the periphery, about 0.1% crosses the blood-brain barrier, gets into the central compartment, and it binds A beta there. Now if you advance it one more time, that increases the clearance of A beta out of the brain. We hope, this is the cartoon again, it shifts these equilibria, and I'll show you some actual data on that, and then that reverses the underlying pathologic process. So that's the cartoon behind what we believe the mechanism to be. So if you go to the next slide, so we do have real data. Now this is a transgenic mouse study, and as you can see on the left side of the slide there, we cured the transgenic mice because they have a lot less plaque when they're treated with this monoclonal antibody. Those things obviously don't always translate into humans. But if you go to the next slide, one of the things we did in phase 1 is we wanted to really nail down the fact that we had target engagement at the doses we were using. And so on the left-hand side of that slide, you see the mouse data. This is plasma A beta, so it's bound to the antibody. It actually goes up. And we saw essentially the same thing in humans. And so that was a good step. If you go to the next slide, this is in phase 2. This is a log scale. So major increases in plasma A beta, not attenuated with multiple doses. If you go to the next slide, so in spinal fluid, it gets a little bit more complicated. If you look at the top 2 plots there, you see the total amount of A beta that's bound and actually unbound. So 0.1% of the antibody crosses, gets into the brain. You see this nice dose-dependent increase in A beta bound to the antibody. If you look at the bottom left on that slide, for A beta 1 to 40, the free A beta 1 to 40, as you might expect, as the bound A beta goes up, the free A beta goes down, and we saw pretty much what we expected. The free A beta 1 to 42 on the lower right, it went up. And the initial question is, why is that? Well, these amyloid plaques in the brain are made of A beta 1 to 42, not so much A beta 1 to 40. So that plaque in the brain is a reservoir, that A beta 1 to 42. And so this was actually one of the first pieces of data to say, you know, we shifted the equilibria enough that those plaques are going back into solution. Normally, your A beta 1 to 42 in Alzheimer's disease in your spinal fluid is low. So this was just returning it back to where you would see it about normally. So if you go to the next slide. Now, I'm going to go through this in the interest of time pretty quickly, but it gets even more complicated, but it also tells us a lot of what we've learned. I mentioned these plaques were A beta 1 to 42, and that's a big part of it. But as it turns out, if you hit it again, the N-terminal end of the A beta is truncated in a lot of cases. And so you see all these different types of A beta that are present. These are in brains of humans with Alzheimer's disease. So if you go to the next slide, so we found in some of our data that we started to see what looked like these N-terminal truncated forms of A beta in the blood after you gave solanezumab. You can't see them before solanezumab treatment, but afterwards it looked like you had bits of plaque coming out into the blood. It took a long time for Ron D'Amatos and his lab to identify these. If you hit it again, it now appears that those fragments are 4 to 42, 11 to 42, and 5 to 42. So if you go to the next slide, and why don't you just hit it again and again and one more time, I think. One more time. So as it turns out, there's all these different types of fragments of A beta that are in the blood after solanezumab. Again, the only one you can measure before solanezumab is 1 to 42. The other ones you have to give the drug to shift the equilibria to find these bits out in the plasma. And there's good literature actually to suggest that this N3pG A beta 1 to 42 is only found in plaque. And in fact, we have another antibody that's now in phase 1 that targets that plaque-specific form of A beta. So we've learned a lot along the way. If you go to the next slide. So with that kind of data to say, yeah, at the doses we wanted to take into phase 3, it got into the brain, it shifted equilibria. We actually did a lot of things that even though solanezumab doesn't bind to the plaque, you actually saw, because of these equilibria shifts, bits of the plaque coming out into the blood. We went into these phase 3 trials. So large trials, as you heard. Let me just, to be fair, I really do have to point out, and you already heard this, we had 2 identical studies that both failed to hit their primary outcomes. Now, when we look at the left-hand part of the slide, that's a secondary analysis. It's the mild patients in the first study that completed, and that was significant, but it was a secondary outcome. On the right-hand side of that slide, we changed our analysis plan. That was the primary outcome, and we just missed. But the results look really similar, and the studies were not powered to just look at that mild subgroup. So if you go to the next slide, so if you take both of those identical studies, combine the mild patients together, here's what we think, what we hope was actually going on from a biological standpoint. And so what we saw is a 34% slowing in the rate of cognitive decline and a 18% slowing in the rate of functional decline, both of those meeting nominal statistical significance, but they were secondary outcomes. Now that 34%, and this question has come up and it comes up more and more, what does that really mean? One way to look at that is that during the 18 months of treatment, you save 6 months worth of progression, and maybe there will be questions for that or during the panel discussion. If you go to the next slide, the safety actually did look pretty good. We had the vasogenic edema for solanezumab was 1% in the drug treated and about 0.5% in the placebo treated. The only things that were statistically significant here actually looked better for solanezumab or worse for placebo. We did see some numeric differences that Rochelle mentioned in some cardiac events. We're not quite sure about that, but we're continuing to follow those very carefully. Go to the next slide. So what else did we learn? Well, one of the things that we did in these solanezumab expedition studies is a subgroup of patients actually had PET scans or spinal fluid to tell us whether or not they actually had plaques, because as these studies were ongoing and about this period of time, it was becoming more and more clear that even the best clinicians would misdiagnose people. And what we found in these solanezumab expedition studies in the patients with mild dementia, about 25% of the time, people were misdiagnosed. And so these are plotted a little bit differently because of small sample sizes, but on the left side, you see all the patients. In the middle panel, those are the people who are amyloid positive, and on the right-hand panel, those are the people who are amyloid negative. So if you just look at the placebo groups, just the blues, you can see if you're amyloid negative, you just don't progress. If you're amyloid positive, the middle panel, you start worse and progress at a greater rate or do progress. And so we know that in the best of hands, if you're doing a study of mild Alzheimer's disease, your clinical diagnosis alone is going to be wrong about 25% of the time. So if you go to the next slide. So based on all this data, we're taking one more shot at this, a third time's a charm, hopefully. So we have the Expedition 3 study that started, and in fact, we just passed the 50% point in our enrollment in that study. So we've applied what we've learned to the design of this Expedition 3 study. It's larger, so we have the right statistical powering. It's only the mild patients, mini-mental scores of 20 to 26. And importantly, one of the inclusion-exclusion criteria is you have to be amyloid positive. We have to know that we have the right diagnosis. And actually, as we look at these screening data in Expedition 3, we're finding the same thing, is that about 25% of people with a clinical diagnosis of Alzheimer's disease don't have Alzheimer's disease. And so those people will not be included in the Expedition 3 trial because the drug obviously targets amyloid. If you go to the next slide. So let me just switch gears here a little bit and talk about something that Rochelle already brought up also, and that's this shift to earlier and earlier patients. So I mentioned that there's this what we're calling preclinical Alzheimer's disease group, people who have plaques but don't have any cognitive symptoms. Now, we at Lilly are involved in a public-private partnership, so there's NIH funding, philanthropic funding, Lilly funding, to do a study called the A4 study, Anti-Amyloid Therapy in Asymptomatic Alzheimer's Disease. In short, these are people who have amyloid plaques but no cognitive impairment. And so we've really shifted much earlier here in the process, way up from even mild dementia, where we are in our other studies, all the way up to the beginning of the disease process for this A4 population. So if you go to the next slide. So it's a pretty complicated screening process for this study. I won't go into all the details, but if you look at people 65 or say 70 and above, about 1 out of 3 will have amyloid plaques, even if you don't have any symptoms. But it's going to be a huge screening effort to get 1,000 people who do have plaques but are cognitively normal. There's also a really interesting bioethics piece to this, is what's the effects of telling somebody who's clinically normal that you've got plaque in your brain, and more than likely you're going to cognitively decline. So that's actually one of the major focuses of the study. So if you go to the next slide, why don't you hit it again and one more time. So this is back to our continuum, and based on our Solanezumab data, and the crenezumab data are actually very interesting, look very similar. It would appear that for an amyloid-related therapy that mild Alzheimer's dementia really may not be too late, that we can slow down the rate of progression even at that point in the disease pathology. But we want to move earlier, and so we're involved with this A4 study. The Diane study I didn't mention, it's relatively rare patients with autosomal dominant mutations that we're also looking at in a randomized controlled trial. And every day we have a discussion about how to fill in with these prodromal patients or MCI. So this is where I think we are. I think we have some promising data. I know a lot of times you read in the press, it's kind of gloom and doom and nothing works and what are we going to do? I think we have some promising data from Solanezumab and now crenezumab that we're on the right track, and that the amyloid hypothesis is a good one. It's a good target. We do have to move earlier in the disease, but again, this becomes an opportunity really to preserve health as much as treat symptoms. And so with that and the bell, I'll close. Thanks.